So good morning once again. My name is Catherine, as I had mentioned, and we are working closely with Brani. And what my presentation will be about is really what research tells us about Phylloxera. And I've been working with Phylloxera for 3 years now. I've been working with Kevin Powell, who is actually who has been known to be the guru of Phylloxera. He's worked with Phylloxera for over 18 years, and I worked with him for 3 years before he got another position with the Sugar Research Institute. And most of this work, as Greg mentioned, to develop procedures and policies, science is very important because most of the policies are based on what works. And to investigate whether a procedure works, it needs to go through a rigorous scientific process. And that's actually most of what that is what I'm going to talk about. What is grape Phylloxera? So Greg has talked about really the importance of the wine industry, and Phylloxera is one of the world's most dangerous enemy for grape Phylloxera. And it is a pale yellow sap-sucking insect, and it is not an aphid, but it is related to aphids. And Phylloxera, just like many insects, goes through different stages. So it goes through the 4 developmental stages: egg, adult, egg to adult, and in between it goes through an incomplete metamorphosis. So what we find is that it goes through different instars, which we call intermediates. We are going to refer to them as intermediates. And when you look at this picture here, that is an adult, and this is an egg, and an egg is about 0.5 millimeters. An adult is 1 millimeter in size, and an intermediate is about that's wrong. That should be about from 0.7 to about 0.9 millimeters. So what Brani and I do in the field, and most of the work actually, Brani is looking at maintaining different colonies in the lab, and we have to investigate them under the microscope, because on naked eye you can only see a pale yellow substance on the roots. You really can't see them with your eyes. You will need a magnifying glass to do that, and that's actually what we're going to be doing in the field. And what you see in the field is, as Greg mentioned, the insects feed on the roots, and the eggs can lay between 200 to 400 eggs. An adult can lay between 200 to 400 eggs. And the crawlers, this is what we call a crawler, and it's very active. So the first time I actually worked with Phylloxera, I went to bed feeling like something was itching, because they walk so fast, and that's the most dispersive stage in the vineyards. They are very active in summer, and that is why the severance period is in summer, because in winter they undergo diapause, so they are sleeping in winter, and then they wake up in summer because they are triggered by temperature. And the crawlers are the most active. They are the ones that are going to go in between from the soil and walk along the vine. So there's potential to be carried in the shoes, on clothes, and the crawlers will be looking for a suitable feeding site. And in the next slide, I'll show you what happens. I'll explain how they feed and what they are looking for as the crawlers are feeding. So, and why grape Phylloxera? So apart from the economic loss, so what grape Phylloxera does is that it feeds on roots and leaves of grapevines. In Australia, the Phylloxera feeds on roots, and that's the predominant stage. But we also find leaf-galling Phylloxera in the King Valley. There's some that have been collected recently. And what they do, the consequences are that they cause the roots to deform, causing nodosities. And this is how the nodosities look like. So, and we hope to find some of those in the field today. So you'll find that in the younger roots, they're going to form little yellow galls, which we call nodosities. And in older roots, they are going to form tuberosities. And that's what we call a tuberosity. We are going to find a thickened, an unusually thickened root. And that is later in summer. That's predominantly what you're going to find. But early in summer, at this stage, what you'll find in the field will mostly be nodosities. So it's good to be watchful on that when you go for the surveillance techniques. Like at this time, really, what you look for will be nodosities, and later on, you're going to find tuberosities. And then because of their feeding habits, they expose the roots to fungal infections, and that can spread depending on other diseases. And we are very much aware, Greg mentioned about the wet season. We know that most of the fungal infections are transmitted in wet conditions. The fungal spores require moisture to be able to spread, and that's Phylloxera exposes the risk of fungal diseases in vineyards. And the next thing they do is they gradually cut off the nutrients and water flow within the plant, and eventually cause root necrosis and death. And that is what you're going to find after years, or even depending on the Phylloxera strain, and I'm going to explain about it, depending on the Phylloxera feeding on the roots, the rate of damage from forming nodosities to tuberosities to root necrosis depends on the Phylloxera strain that is within the region and how virulent it is in causing death of the vine. Yeah, if you can't, please, and well, because of my accent, just if there's a word that you can't understand, just shoot your hand up and I'm going to explain better. Okay, what do you see in the field? When you get into a vineyard, what do you really look for, or how can you really tell that there's Phylloxera? So one of the things that you're going to find is if you find that there's an unusually big undergrowth under the vines, that would be a potential. It doesn't say that Phylloxera is going to be there, but it could be a hint. You know, finding Phylloxera we found, and Brani will testify, it's like looking for a needle in a haystack. Sometimes it's there, sometimes it's not. In areas where Phylloxera is very dominant, it's easy to just go and say, okay, that vine has Phylloxera. But in other areas, like in the Mornington, where Phylloxera hasn't been found, it's really going to be looking for Phylloxera in a haystack, a needle in a haystack. So you will need some hints to be able to tell where do I go to begin with, because you don't want to spend the whole day digging holes when you really don't have to. So, but these are just some hints before you really narrow down into what you want to look for. So weed undergrowth is one of them. And then stunted growth and small grape bunch sizes will be another big hint. And we have found that in the vineyards that we've gone to. So when you go along the vine, along the rows, you can tell that that's really a healthy vine. But then next to it, you're going to find another vine that is really stunted. And that's really a hint for you to dig. And then the other thing is early yellowing. So you'll find that some of the vines are going to have yellow leaves, and others are really having a very vibrant green. So, and that's also is a very good hint for you to look at. And of course, dead vines. That's really, a dead vine is an absolute hint that Phylloxera could be present. What I didn't mention is that when Phylloxera attacks a vineyard, it takes years for the symptoms to show. So Phylloxera could be present in a vineyard now, for example, but that first season, you can really not tell whether Phylloxera is there. But in the next year, you might see a combination of all these visual symptoms. And in the next year, for example, this is, so the weak spots, Greg mentioned about the weak spots. So you're going to find that if you, for example, in some of the vineyards that have a very nice view, when you're looking at them, you'll find a spot, like a rectangular spot or whatever that has weak vines, and you can see some of them having a yellow characteristic, and the vines that are surrounding them would have a very vibrant color. So and that's really what you look for. So those weak spots would be a very sure hint that you would need to go and look there and then before you go into the other healthier vines. So in the King Valley, in 2000, that's how that vineyard looked like, and 3 years later, that's how it looked like. So you can see that it takes time for the symptoms to show, and that's why Greg mentioned that this is going to be a 3-year surveillance period. So you would be able to note, if there is Phylloxera in the 3rd year, you would be able to note the differences, because some of the, depending on the genotype of Phylloxera, you will find that the feeding habits of it will be more vigorous and causing more damage compared to other genotypes. So the thing to watch for are the weak spots and the yellowing of leaves, and eventually what causes the death of the vine is that gradual fading and exposure to and cutting off of the nutrients on the vine. Let me see whether I have... Oh, before I go to management, let me talk about, I didn't put a slide on this, where in the northeast where we are in Rutherglen, Phylloxera in around 2000, a scientific study discovered that Phylloxera exists as 83 biotypes. What that means, it's like tribes. So where I come from, we have different tribes. So we are the same people, but different tribes. So let's look at them as tribes. So Phylloxera has 83 tribes. And each one of the and we'll call them biotypes. Some of the biotypes are very virulent. That means they are going to feed more on the plant, causing more damage, where others are really, we can call them harmless, but they will still cause some harm. So there's a degree to which Phylloxera can cause damage on the plant, and it depends on their virulence characteristics. In this area, we have the genotype called G1, which is pretty virulent. We find in the lab that when we because we maintain these different biotypes in the lab, and we find that one of the genotypes will reproduce very, very quickly, will produce masses and masses of eggs. And that means that we're going to find it, the eggs will develop into the crawlers and they'll spread out, and each generation is going to multiply very quickly. And what that means is that each genotype, depending on the genotype that you find, the damage is going to be much, much more quicker. So that is left for the taxonomists to find out, like within the next 3 years, should you find Phylloxera. So how will that change if you find Phylloxera in the first year? How will that translate when you come to the end of the survey? So it's good to be watching out, to be careful, like when you see that there's a difference in a vine characteristic this year and the next year, then it's good to be watchful, because in the 3rd year, it could happen like what happened in the King Valley. So that was another virulent genotype. Within 3 years, the vines were actually almost dying. All right, how do we manage Phylloxera? So there's no cure for Phylloxera, as Greg has mentioned. There hasn't been a cure as yet, and that's why research is important, because one day we hope to have a cure, but at the moment we do not have. The only way to manage Phylloxera is to contain it and ensure that it doesn't spread. So we need to manage the risk so that it doesn't spread to areas where it doesn't need to be. So at the moment, the integrated management is through quarantine, detection, and host resistance. And what we mean by that, with quarantine, and that's really where we come in. I mentioned about biotypes. I don't know whether this is too small, but this is Victoria, and where we have the red dots, that's where we find Phylloxera. In North Victoria, around here, we have all the 83 biotypes of Phylloxera. In the Yarra Valley, down at this section, we have G1, and what we try to do with quarantine is to contain Phylloxera where it's present and minimize its risk of spreading to other areas. And there's a national Phylloxera management protocol, and that's what Brani and myself have been doing in looking at, has there been a quarantine breakdown? We need to validate that these protocols are really working. And for the past 3 years, that's what we have been doing, validating that these protocols that are in the national management protocol, that they are really working. And so that when there's movement of material out of a PIZ, which is a Phylloxera infested zone, that the risk of transmitting the Phylloxera to other regions is minimized. And the quarantine measures include using disinfestation techniques, using heat treatments or chemical treatments. And this, we have found through this, from the past 3 years, that different genotypes respond differently to heat treatments and chemical treatments, which really has been a very worrying outcome, let's say, because we expect that when you treat an insect, it's going to be killed, but we found that it's not really the same. You can treat the insect, some will die, some will not, which now creates another dimension that we need to be very, very careful to use the most rigorous method when it comes to disinfestation techniques. Then detection is the other method, which is actually most of what you will be involved with, because the first thing to solve a problem is to detect that you have a problem. And there have been different ways of detecting Phylloxera. Digging is one of them, which is what we're going to do now, and digging is mostly used during the surveillance techniques. And then using DNA probes, where we go into the field, take a DNA sample in the soil, and take it to the lab at AgriBio, as Greg mentioned, and that really identifies whether the sample you have is Phylloxera. And then the next one is using EM38, which is soil conductivity. Research has shown that different soils that have Phylloxera, or roots that have Phylloxera, the soil electric conductivity is different. And there's a technique which is called EM38 that can be used to detect Phylloxera that way. And then the chemistry, leaf chemistry has been one of them. And trapping is actually what we do if we want to investigate what Phylloxera is doing over the season, and we have a lot of that work going on. So at the moment, what you're going to be dealing with mostly will be digging. And then Greg did mention that, and the only way to actually manage Phylloxera is through replanting with rootstocks. But that means that already, whatever you have, most of the Australian vines are on roots, and we find that on roots is really very susceptible to Phylloxera. So Phylloxera came from America, and in America, Phylloxera is resistant to those roots there. But when Europe got their roots, when Europe got rootstocks in Europe, where most of our rootstocks come from Europe, are very, very susceptible to Phylloxera. So what we try to do, or what has worked, is going back to America and looking for those resistant rootstocks And there is a big trial going on with CSIRO and in collaboration with us to test different rootstocks that are resistant to Phylloxera. But having said that, that's a long-term project because you have to wait for the vines to die before, as Greg mentioned, before you can replant. So that costs money to the grower, and it takes to replant in a vineyard, it costs $25,000 to $50,000 $25,000 to $50,000 per hectare to replant on resistant rootstocks. But also, having said that, we have a trial that is going on in the King Valley where we have identified Phylloxera developing on rootstocks. So that creates another question about this insect because it's really, so it means that some rootstocks will be susceptible to Phylloxera even if they are rootstocks. So is that too confusing? I hope that's okay.
Well, are the rootstocks that you're finding Phylloxera developing on, are they able to survive with Phylloxera, whereas the unrooted plants would die anyway?
Yeah.
So they can sort of live together, but it's not ideal.
It's not ideal, exactly, because they're tolerant. So, and that means if they're tolerant, then they can be transferred. So it may not be developing, it may not be harming the vine, but it's a risk because we are working in the vineyard and the risk of transfer is very, very high. It still poses a big risk. Yeah.
So there's no systemic insecticide or soil drench that can be used?
No.
That doesn't wipe out your vines as well.
Exactly, and I think it would be a very it wouldn't be very cost effective because this insect lives in the soil, so you would have to put in a lot of insecticide for it to die. So, and since vines are in a big acreage, that really wouldn't be a very cost effective. And in the studies that we've and in the literature, actually even in America and in Europe, the insecticide hasn't been one of the things that they use. Yeah, so it's really just management. Yeah. Okay, I think I would like to perhaps, if anyone has more questions in this, because this is really where you come in, and that's where a lot of information in your head needs to go to at this time. If you have any questions or discussions at this time before I move on to the next one, that would be good. Any questions so far? All right, good. So the next one is clothing. As I mentioned earlier, you in summer, especially come late summer in January and February, that's when the insect is really more active. The generations. So since spring, Phylloxera has been reproducing. In summer, they will have exponentially increased. So you're going to have tons and tons of Phylloxera, especially where the infestations are. And what happens is at that time, they will be competing for feeding sites. And if they can't find the feeding sites, they're going to start moving, and you're going to find them on the leaves. And that's the potential of getting on your clothes is very, very high. So how do we disinfest clothing? So we looked at different options, and there are two options that work. So Mortin Doom is something that you're going to put in your shopping list because that has been shown to kill Phylloxera. But with the condition, you have to spray the surface of the cloth for 5 seconds. So that has been shown to kill Phylloxera. Or I believe you're going to be wearing suits, the white suits, when you go into the vineyards. Dispose of those suits and put them in a bag, and that needs to be treated and disposed in the right manner. And if you do not have Mortin and you don't have white suits, then you need to put the cloth in a bag and take it home. I think this would not be a very good option. Greg, I don't know what you've planned, but if you don't have the suits and you don't have Mortin, then you need to bag to put the clothes that you have in a bag and wash them in a hot wash cycle for 30 minutes. So that,
Our process will be rotating overalls. We do carry more things and use more things, the hats as well as the overalls.
yep,
And then we put them in black solar bags and leave them on the vehicle as we rotate them. So if we're doing another video, for instance, these mortaring bags in solar conditions on top of the motor car in the sun. We found that effective.
okay,
I can't prove Phylloxera, but Phylloxera turned up where we were being.
yeah,
But the solar bags, we found that very, very helpful to kill anything.
oh, all right.
As
So,
well as more too,
yeah,
on your clothing and hats.
okay,
Because it's getting above 60 degrees.
yep, yep. So has that been tested with a thermometer? That it,
Yes.
yeah, as long as it goes below 50 degrees, we've shown that all temperatures of 50 degrees for at least 1 minute will kill Phylloxera. So as long as that temperature is above 50 degrees. Yeah. Okay, so I think that's really all because for shoes and clothing, but I'll quickly go through the other heat disinfestation procedures for harvesting beans. And one of the things so I think you wouldn't be doing this because it's really for harvesters. So the treatment is dipping beans in water for 70 degrees for 2 minutes, but we have found that 50 degrees works, so that will save the growers some money by reducing the temperature. So, but all we do is do the research, and then there are people who are going to develop the protocol and amend it appropriately. Tractors and vehicles, you might sometimes be required to do this when you get into a property because you're going to be driving in and out. Steam cleaning is effective as long as steam contacts the surface of the tires and for 10 seconds. It's got to be 10 seconds. And the farther you move away from, the steam source moves away from the tires, the lower the temperature. So caution must be taken to ensure that steam is as close to the surface as possible. Viticulture machinery, this is where tractors and harvesters are used. So the tractors need to be subjected into a heat shed that is heated to 45 degrees for 75 minutes, or a heat shed that is heated to 40 degrees for 135 minutes. So the root bundles will be held in water for 50 or 54 degrees for 5 minutes. So you won't be required to do that. And for diagnostic samples, these are samples that I think you won't be required to do that, but freezing, we found, or research has shown that freezing at minus 20 for a minimum of 12 hours will kill Phylloxera. So it's got to be minus 20. So Kevin did get word that 4 degrees kills, but we found in the lab that 4 degrees does not work. So it's got to be minus 20 degrees. Yeah, which is freezer temperature. Not in the fridge, the freezer. Yeah, the fridge will not. They're very happy. They're very happy in the fridge. It just gives them a cold, just a bit of a swim or in hot summer. And that's really all we had. So if you have any questions.
Is there anything else that actually affects, or is it just grapes?
No, it's specific to grapes. So that actually it's called the grape Phylloxera. So it only attacks grapes.
You mentioned the period between disinfesting your shoes and then not instantly rinsing with water. Do you guys have like a timeline that the contracts can sort of say, oh, after 10 minutes you can probably give them a bit of a rinse? Or is there
We
just,
actually don't.
No.
you don't rinse them at all?
No.
It's not, so it does wet. Yeah, it's going to wet your clothes. You end up with purple clothes if you do splash, and Brendan will be able to back this up. Wet your shoes, all of that.
Yep.
But there is no other, and that's what our work is, trying to find something that is an alternative to that. At the moment, there is not one, but we're hoping to find something that's a little bit user-friendly for that reason. But no, ideally, and it will rust out with spades, it will do all that, but that's the only way we can do it.
Yeah.
But the trick is to, if you've got bleach and you've got spades and you put them back in the vehicle, is to put them in a garbage bag and seal it so you reduce that liberation of the smell. But, yeah, you end up with a headache by the time we get back to our other Glen on a 3-hour trip.
Yeah. Yeah. So I think that's why I mentioned maybe consider having 2 pairs of shoes. One that you are happy to throw away after this season, and because they do wreck your shoes, and yeah. I think it's recommended because, Brani and I will testify. We go home with patches of bleach, and you wash them, and the next time they're all
What we
bleached
have
out.
to do, and our protocol is, every new vineyard, change of clothes. We wear a full suit every time, but and when we go home, wash our hair, all of that before we go into another vineyard. With us girls, a bit longer hair, it's worse, but it doesn't matter. Any short hair, they'll still only take that one insect.
Yeah.
Avoid contact with blinds at all times.
You guys are going to have that extra hassle of probably nets as well when you're in the vineyard as well.
So the risk is high.
Dealing with nets. And that actually comes from the shovel.
Yeah.
Yeah.
Least contact with anything in the environment
Yeah.
other than boots and shovels.
Yeah.
That's it with us. Don't touch the lines, don't need to. Cleaning process we've gone through and increased it to 60 seconds, which I think is fantastic.
Yeah.
And with the rotation, we're usually pretty tight.
Yeah, excellent.
Cleaning areas are in usually areas like that out there, all 14 mil bluestone.
Yeah.
It gets very hot. We don't move until all our boots and shovels are dry from the flooring. So, that's something we've learned in the time together with you. So.
Yeah, that's good. And
Because the thing is, you guys, it's really important because if something does happen, and it will most likely not be from you guys at all, but if something happens on that vineyard, the department or Brendan, that's where the finger will be pointed. So prove that he's ticked off everything properly.
Yeah.
It's really important. So you guys have to support him to make sure that that's done. And I'll be guaranteed it won't be you guys if something does happen, but it'll be the first thing that will be pointed out.
Yeah.
I'm very strict on this program.
The other thing to consider is restricting yourself to an area. Like, if you don't have to go to another vineyard in a day, just do a vineyard per day, or a close vineyard rather than going from here to like 20 kilometers away. So restricting yourself to an area. And this also goes from broad to narrow. When you come into a vineyard, restricting yourself. If a vineyard is infested, just walk, get to know where you come from, what area has Phylloxera, and avoid going to an area that doesn't have Phylloxera when you're disinfesting. So, I think maybe if that is the wall, if beyond the wall that's where Phylloxera is, then you disinfest your boots out there and come in here and not go back. So it's really trying to avoid as much contamination as possible. Yeah.
And probably another point too is when you're going out looking, and Catherine said that weak spot, sometimes another term you might hear on the tracks is an oil spot. That's how they explain it, because it sort of starts in the middle and then spreads out. The trick with you guys, if you're going in there, and that's where you're going to go to first before you start the surveillance, like every third row, fifth kennel. But before that, if you see it, you'll head there. But you won't go into the centre of that oil spot or that weak spot, because the Phylloxera populations have got so big that's why the plant started to die, it started to yellow, that going to be moving, they've moved out further to look for nice, they like the nice sappy new growth. That's what they feed on, that's what they populate with.
We call that
So,
the
yep.
we call that the two bob theory on the billiard table.
Yep.
It's spreads.
It's spread. So you're going to go just outside. So you're going to see a weakened vine. So it's yellowing, it's not as much growth. You'll go to the next vine next to it because that's where they, even though you mightn't see, as Catherine said, it can take up to 3 years depending on soil type, all of that, for you to visually see the differences between 1 and 3 years. But in the roots, they might be there, so that's where you would look first. That's where the numbers will be. If you move into the food source, so don't worry about where it's dead, there'll be no food source left there. Go to the phenotype. Save your digging.
Yes.
That's all where it's going to be. Kill them.
The first 2 hours of the first being out of the
Yeah.
And go to find it. I mean, you probably think, you know, it's not here. You're there to find it. Yeah, that's your job. There's snip dogs.
Yeah. All right. Thanks, Greg. Yeah.
Thanks.